Hi, my name is Evelyne Tantry, and I'm a postbac from the lab of Dr. Benjamin Arenkil at the Baylor College of Medicine. Today, I'll be presenting on how olfactory-guided behavior evokes distinct neural patterns in parvalbumin and somatostatin GABAergic basal forebrain neurons. So when we talk about addiction, one of our first thoughts are substance addictions, such as drugs or alcohol. More recently, however, we have begun to also recognize behavioral addictions, such as gambling or kleptomania. Despite their unique features, all these addictions have in common that they're characterized by pathological reward seeking, in which there is a compulsive desire to engage in activities that provide the individual with a rewarding stimuli. Thus, it becomes imperative to elucidate the neural mechanisms that regulate addiction and addictive behaviors, beginning with sensory perception. So presently, little is actually known about how sensory stimuli is encoded in the brain. And to study sensory perception, we must take into consideration the variable behavioral conditions and contexts that shape it. This thus makes the basal forebrain, a known hub of behavioral state regulation, a prime region of interest. Within the basal forebrain is a dense population of cholinergic and GABAergic neurons that project to the olfactory bulb. Thus, for our experiments, we utilize the mouse olfactory system as a model for study. Additionally, a paper by Xu et al. Reveals that among the GABAergic neurons that project from the basal forebrain, the somatostatin and the parvalbumin subtypes have differential effects on regulating arousal, suggesting to us that these GABAergic populations may also differentially regulate decision-making and therefore reward-seeking. Thus, bearing in mind these cells' potential role in modulating a myriad of complex behavioral functions, we decided to focus on studying local GABAergic signaling within the basal forebrain. To this end, we coupled pyrophotometry with an olfactory-guided go-no-go discrimination task to observe GABAergic activity in mice, coupled with odor-guided decision-making behaviors. So for those of us who aren't too familiar with it, a go-no-go task requires a water-restricted mouse to undergo training such that they may know to receive either a positive go odor or a negative no-go odor from the odor port here shown in white. These odors serve as a stimuli that they must learn to associate with either receiving a water reward or not receiving a water reward from the reward port here shown in blue. For a trial to be counted as a hit, the mouse must recognize the go odor and seek a reward, in a timely fashion, of course. Failing to seek a reward, however, constitutes a miss. When a mouse receives the no-go odor and knows not to seek a reward, the trial is considered a correct reject, whereas when they try to seek a reward despite receiving a no-go odor and they're not going to receive a reward, this will constitute a false alarm. This assay essentially allows us to be able to determine decision bias as well as reward seeking, all while we are able to couple it with pyrophotometry so that we can also pair this with in vivo imaging and therefore correlate these behaviors with the appropriate neuronal activity. So our group has previously shown that recording neural activity from a general population of GABAergic basal forebrain neurons reveals a spike in activity during odor port entry and a suppression during reward delivery. But to verify that GABAergic signaling is indeed at play in basal forebrain activity, we incorporated a chemogenetic tool known as designer receptors exclusively activated by designer drugs, or DREADs for short. And we incorporated this to the go-no-go assay, which enables us to suppress all GABAergic signaling. So compared to a control cohort who only administered saline, the CNO-infused DREAD cohorts had decreased decision bias, or in other words, they were less likely to seek out rewards, suggesting to us that GABAergic neurons may be involved in modulating reward-seeking behavior. However, because we know that somatostatin and parvalbumin GABAergic neurons serve opposite roles, it is necessary to parse out their specific involvement in modulating decision-making. Thus, we recorded from parvalbumin neurons genetically expressing a calcium indicator during go-no-go. We saw that when we quantified the mouse's odor and reward port entry, and temporally aligned all of this data with PV neural activity, we saw that activation of these PV neurons during odor port entry was spiked, while there was a strong suppression when there was reward delivery. And this pattern is consistent with what we saw earlier with the non-specific GABAergic activity, which suggests to us that PV neurons constitute a sizable percent of the GABAergic signaling we saw before in the basal forebrain. Likewise, when we measured the peak signal amplitude following odor presentation, we saw that there was no difference between the trial type averages. However, when comparing the area under the curve after a reward was sought, we saw that there was significantly decreased activity when a reward was actually given during the HIT trials, which suggests to us or supports to us the finding that PV neurons are suppressed during reward delivery. Conversely, recording from somatostatin neurons showed us that there was initial similarities in the spike during odor delivery. However, there is a divergence when there is the reward delivery, as there was actually no significant decrease when the mouse receives a reward. And when we measured the peak signal amplitudes during odor presentation, there was also no significant difference between trial types. However, when we compared the area under the curve during reward delivery, we saw that there was actually an increase in signaling as opposed to when there was no reward being given. So again, this shows to us that somatostatin neurons actually don't show any notable decrease, but might even have an increase in signaling when a reward is given. So it should be noted, however, that the magnitude of somatostatin activity immediately following reward port entry was not found to be correlated with decision bias or accuracy when the rewards were sought. However, odor-evoked activity in the somatostatin neurons are correlated with decision bias and odor discrimination, as seen here, with given the significant correlation between criteria, which is decision bias, and sensitivity, or D prime. This indicates that the activity of somatostatin neurons in the basal forebrain may predict the success of mice in discriminating odors, but not when they're seeking reward. Meanwhile, for PB neurons, there was no significant correlation between either behavioral parameter. So in conclusion, we found that basal forebrain neurons played a role in regulating decision bias and accuracy. Specifically, we found that PB expressing GABAergic neurons in the basal forebrain are activated during odor presentation and suppressed during reward delivery, whereas somatostatin expressing GABAergic neurons are activated during both odor presentation and reward delivery. Somatostatin neurons also exhibit a strong correlation to task performance and decision bias. This data suggests that the subtypes of GABAergic basal forebrain neurons are differentially involved in decision-making, and that their specific bidirectional activity may influence olfactory discrimination and decision-making. I would like to thank my PI, Dr. Benjamin Arenkel, my mentor, Dr. Hansen Moss, and the Arenkel Lab for their support and input on this project. Please feel free to reach out to me with any questions. Thank you so much for your time.